This conference will now be recorded.
Good evening, students. I'm Dr. Divya from TeenEinstein. So we are into the second chapter of 12th biology, right?
Good evening,
Sexual
ma'am.
reproduction in flowering. Good evening. Sexual reproduction in flowering plants, right? In the last class, we have discussed about different pollinating agents, abiotic and biotic pollinating agents, right? So today we are going to start with outbreeding devices. So now we reached up to pollination, right? The pollen grain, it is reaching on stigmatic surface. So pollination is done. Next is outbreeding devices. Okay. So outbreeding devices. What do you mean by outbreeding devices? Devices means what actually? It's a plan or it's a thing or method, right? It's a method to achieve a particular aim. That is what called as device. We used to tell like that device is not working, this device is not working. So it's a kind of mechanism or a plan to achieve a particular aim, right? So what is outbreeding? Then next we are learning about outbreeding devices. So devices means it's a method for a particular thing to be done, right? So outbreeding, what do you mean by outbreeding? Outbreeding means mating or breeding between individuals from different populations or different species. Got it? So outbreeding means it's the exact opposite word of inbreeding. Inbreeding means breeding or mating of the organism between closely related species or related by ancestry. You know that that ancestral group, those group will be closely related. There will be similar characters, everything will be present. The almost similar characters or that group of organisms will be having particular characters. Okay, that will be almost similar. So inbreeding means the mating of organisms which are closely related. Those organisms will be closely related. But the outbreeding, what do you mean by outbreeding? Outbreeding is the exact opposite thing. Means there is no close relation. Okay. That means mating or breeding between individuals from different populations. See, one population will be having, or population means a group of organisms that will be having similar characters. So inbreeding means within the population that mating is happening. So outbreeding means in between different populations. Okay, it's not between the individuals of same population. It is between different populations or between different species. That is called outbreeding. Out of the population. The breeding is taking place out of that particular population or a species. So what is the aim of this biological breeding? Why we are doing this breeding? Or biologically, what is the importance of that breeding? What is the importance? To create diversity or difference. See, breeding in between the same organism means there won't be any change in their characters or there won't be any development in that organism, right? So we need some difference or diversity in between organisms. So diversity will be created only when cross-pollination or outbreeding happens. Only during that time. Outbreeding means, just now I said outbreeding. What is outbreeding? Mating or breeding between individuals of different population. If individuals of different population are getting into a breeding program, what will happen there? Different characters are coming into the next individual or the hybrid. It is coming to the hybrid. Different characters which are from the one population and some important characters from another population. Both characters are getting mixed in the hybrid or the next generation. So outbreeding brings more diversity or Cross-pollination brings more diversity. So biology encourages outbreeding, right? Then only we will get more diverse organism, more diverse plant species and all, right? So in the case of plants, they produce hermaphrodite flower. Hermaphrodite, what is the meaning of that hermaphrodite, that term? That means both kind of reproductive organism, or that can, both kind of reproductive organs, or they will produce both male and female gametes. So if the male and female gametes, they are in the same flower, that means pollen grains of one flower can come in contact with the stigma of the same flower. There is a chance of self-pollination. Usually biology encourages cross-pollination, then only diversity comes, right? So if in the case of flowers, they will be having anther and stigma both on the same flower. In the same flower, both are present. See, you can see in these flowers, anther is there, even stigma is also present. So both male and female organs are there in the same flower. So there is a chance of self-pollination. From the same, this flower only, this anther will go and reach on the stigmatic surface. So there is no cross-pollination, there is self-pollination. Again, similar kind of plants will be produced in the next generation also, right? But this kind of continuous self-pollination, it will result in the inbreeding depression. What is that? Inbreeding depression. So inbreeding means within the population only that breeding is happening. So that will create some kind of depression because there is no genetic changes. That means those organisms will slowly lose the fitness. Fitness to what? Fitness for what? They will lose the fitness for survival. Right? See, everything is getting changed. So they should get adapted to survive. For the survival, they need more adaptations or more evolved characters from the previous one, right? So they need some kind of changes. So there is no difference in the character genetically in the case of inbreeding organism, right? So we need to create a degree of changes to the next generation that should get increased in the next generation. Otherwise, inbreeding depression will come. So what is that? Outbreeding device means the mechanism or the process in which the plant adapt to prevent self-pollination. The plant is getting adapted to prevent the self-pollination. Why? Because that need to create some changes. There should be some genetical changes in the next generation. So plant itself creating some kind of mechanism or process with which they can adapt to prevent self-pollination. They want to prevent the self-pollination. And self-pollination may cause inbreeding depression. Self-pollination again and again, same plant, it's getting pollinated itself. And there is no pollen grains from outside. So that will create inbreeding depression. Okay, there won't be any kind of genetical changes in that particular species. So it won't be fit for the survival, or slowly it will lose the fitness to survive. So there won't be any kind or any difference in the character. So genetically they will lose that heterozygosity, or genetically the degree of homozygosity will increase. What do you mean by the degree of homozygosity is increasing? Homozygosity means that similar characters. See, if parents are different, they'll get different type of character from one parent and from the other parent. Then in the next generation, there will be a kind of change from the previous generation. But if it is getting pollinated itself, there is only homozygous genes. There won't be any heterozygous condition. Only homozygous genes will be there. So similar characters will be continued generations after generation. So degree of homozygosity will increase. Okay. So the reason behind the adoption of such techniques by plants is that due to continued inbreeding, plants can develop a condition called inbreeding depression. See here, I have clearly written in the slide, it's very clear. Due to the continuous inbreeding, plants can develop a condition called inbreeding depression. So inbreeding depression it will lead to unhealthy offspring. Inbreeding depression means without any changes, all generations are getting produced, means that will lead to unhealthy offspring. So definitely it won't be fit for survival. Okay. For the coming generations, it won't be fit for the survival of that respective organism or the plant. So it cannot survive. There should be some kind of changes in the genes to survive. So due to the increase in the degree of homozygosity, similar genes will be continued to the next generation also. So there won't be any heterozygosity. So it will lose the fitness. Slowly it will lose the fitness. So plant itself is preventing self-pollination to reduce the inbreeding depression. Okay. So let's see what and all plants are doing. So flowering plants have developed several devices or several methods to discourage the self-pollination. Okay. And plants are encouraging close pollination. For what? For genetic diversity or for the fitness or to increase the heterozygosity and to reduce the homozygosity. Plants are producing several or plants are developing several methods or mechanisms to discourage self-pollination and to encourage the cross-pollination. So among that, so outbreeding devices we are going to discuss one by one. So in the first method or mechanism, among this, in some plants, pollen releases and the pollen grains are getting released in one time and the stigma receptivity that are not synchronized. Okay, the first method or first mechanism that plants are adopting is the pollen release and the stigma receptivity are not synchronized. They are not at the same time. There is no synchrony between the stigma receptivity and the release of pollen grain. In this case, the pollen either the pollen will get released before the stigma becomes receptive. Okay. The stigma should be ready for the reception of that pollen grain. That pollen grain should come to that stigmatic surface, but in that state, that stigmatic surface should be ready to receive that pollen grain. Okay. There's some kind of chemical production is there and there is an interaction between the chemicals of this stigmatic surface and the pollen grain. If everything is matching, then only the pollen grain can germinate or the pollen grain can survive on stigmatic surface. Okay. So if the stigmatic surface is not in a condition to receive the pollen grain, then also that is a failure, right? So in some plant species, pollen release and the stigma receptivity are not synchronized. In that case, either that pollen is released before stigma becomes receptive. That pollen will get released before that stigma is getting ready for the receptive purpose of the pollen grain, or the stigma become receptive before the pollen release. The stigma will become ready before that pollen is getting released, before that stigma will be ready. In both cases, there won't be any synchrony. Got it? There won't be any synchrony. There is no synchronization between the pollen release and stigma receptivity. These and all for preventing self-pollination. Plants, they are themselves adopting all these technologies to prevent self-pollination because they are encouraging cross-pollination to reduce the inbreeding depression. So first method, in that flowering plants are developing many devices. In that first one is the plant species, in particular plant species, pollen release and stigma receptivity are not synchronized. That is the first thing. This, not in all plants, some particular species are following this method to prevent the self-pollination. So either the pollen grain will get released in one time and that time the stigma surface won't be ready for or ready to receive this pollen grain. Otherwise, the stigma will be ready, but the pollen grains or the pollen release won't happen at that time. or the pollen grain or the anther won't be ready to dehydrate this pollen grain. Any one case means there is no synchrony between both, means either the stigma or the pollen release. Both are not happening in the same time. Okay. So that is the first method. And in the second method, anther and stigma are placed on different position. Here you can see. Here anther, these are the anthers. And here stigma is here. In the second picture you can see stigma is away from the anther. Here anther you can see in this bunch. Here one bunch of anthers you can see. But the stigma, it is away from this anther. And it is, see here anther, it is not tall, but the stigmatic surface is away from this anther. So in this case also, pollen cannot come in contact with the stigma. From the same flower, if this anther dehiscence is happening, then also this stigmatic surface is away from the anther, right? So in that case also, from anther, these pollen grains cannot reach on the stigmatic surface. Got it? So that's how this type of flowers are preventing self-pollination or self-fertilization. Okay. So in the first method, one of the strategy which is adopted by some plant species include pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive, or the stigma becomes receptive much before the release of pollen. So in the first case, there is no synchrony between pollen release and stigma receptivity. And in the second case, the anther and stigma, they are placed at different positions. The position of anther and stigma, both are different. Both are not at the same level. So that the pollen cannot come in contact with the stigma of the same flower. So in both cases, we can prevent self-pollination. And in this, the method which the plant is adopting is cross-pollination, or the plant prefers cross-pollination in both cases. Okay. And next method, we have listed out around 9 methods in this. So next method is to prevent incompatibility. So in this, the pollen from the same flower that may not be compatible with the stigma of the flower. Got it? Pollen grains. In the third method, we are discussing about self-incompatibility. Self means from the same flower, that pollen grain, if they are reaching also, those pollen grains cannot lead to a fertilization. How? Because they are incompatible. They are not matching. Okay. That means the pollen grains cannot germinate on the stigma. If they are reaching on the stigmatic surface also, they cannot germinate on the stigma because they are not the matching ones. Got it? So the pollen grains from the same flower. See, if the pollen grains in that anther and stigma both are in same level, in all other ways it is a perfect flower. But by chance the pollen grain is reaching on stigmatic surface, then also that pollen grain cannot germinate on the stigmatic surface of that same flower because they are incompatible. They are not matching each other. Okay. So that's how in such type of plant species it is preventing self-pollination. Got it? Next, the fourth method is production of unisexual flowers. Production of unisexual flowers. The another method to prevent self-pollination is the production of unisexual flowers. In this method, both male and female flowers are on same plant. They are on the same plant. But pollens from the same flower to the stigma, it is not possible. From one flower, the pollens are reaching on that stigma and germinating. That is not possible. Okay. But in this case, we can prevent self-pollination or autogamy, but we can't avoid gametogamy. What is gametogamy? Pollen grains from the same flower, some external agent like vector or any pollinator or wind or something. From one flower, the pollen grains are carried to the stigma of another flower, but that flower will be on the same plant. Okay. See, here we said production of unisexual flower. Okay. Unisexual flower means the production of unisexual flower, both male and female flowers are on the same plant. Male flower and female flower both will be on the same plant. But pollens from the same flower to the stigma, that is not possible. Okay. Male flower and female flower both will be present on the same plant, but from one flower to the same stigmatic surface, that pollens are not allowed. Means or it won't work out. But in this case, we can prevent self-pollination. We can prevent self-pollination, but gametogamy we cannot prevent. We cannot avoid that, because from the through any vector or any pollinating agent, or that may be abiotic or biotic, like that, any agent can pollinate the flower. How it will? The pollen grains are carried to the stigmatic surface from one flower, the stigmatic surface of the another flower, but both flowers will be on same plant. So you can see in this plant that pollen grains from this flower may reach to the stigmatic surface of another flower. Not on the same flower, it can reach on the another flower, another stigmatic surface, but both flowers are on the same plant. Got it? It's almost a self-fertilization, but self-fertilization in the sense it's on the same plant, right? It's almost like a self-fertilization, but it is called as gametogamy, right? Examples for this one is maize and castor. Maize and castor, in that unisexual flowers are produced and pollination is happening in the way which I have explained now. And next method. So don't forget the examples for this. For the unisexual flower production, castor and maize. Both are important MCQs. Note down castor and maize. And next, fifth method is male and female flowers are located on different plants. The male flower and female flower both are located on different plants. So the male flowers and female flowers are located on different plants means there is a male as well as a female plant. Okay. For example, you know, in the case of papaya, in papaya, there is no autogamy or gametogamy. Got it? Both gametogamy. What is gametogamy? On it, that another flower, both flowers should be on the same plant. But in the case of papaya, there is no self-pollination and there is no gametogamy. Both, there is a male plant and a female plant. On male plant, there is no female flower, and on female plant, there is no male flower. Okay. So both plants are different. Male plant and female plant both are different in the case of papaya. Got it? So which is the male and female flowers are located on different plants. So example for this case, different male and female plants are separate. That's how the plant is helping for inbreeding, to reduce the inbreeding depression. Example for that is papaya. Important MCQ, papaya. Note down. Papaya is an example for that. And next, sixth method is prepotency. Prepotency. In pollen prepotency, a different flower's pollen grain grows faster on stigma than pollen from the same flower. See, pollen from the same flower means it's a self-fertilization. Again, it's an autogamy, right? So to prevent that, plants are adopting one method, a pollen grain from a different plant, different flower that will grow faster on the stigmatic surface of the flower, another flower. So they are getting a pollen grain. They are giving preference to the pollen grain from another flower. This plant prevents autogamy. That is to prevent the autogamy. So this is also an outbreeding device. An outbreeding device. So prepotency, example for prepotency, apple, pear, then grape. You have to remember these examples. Mainly from this portion you may get these MCQs, important MCQs. So grapes, apple, and pear. These are the examples for pollen prepotency. What is that? Pollen prepotency means a different flower's pollen grain. They are encouraging a stigmatic surface of a flower. It is encouraging the pollen grains from another flower, not from the same flower. That pollen grain will germinate faster than the pollen grains from the same flower. Okay. So that's how it is working as an outbreeding device. Next is suppression of one sex. Seventh method, suppression of one sex. The method, the suppression of one sex, that means in some bisexual flowers, either stamen or carpel, that is completely suppressed and any one will be sterile. Okay, the stamen or in bisexual flowers, in the case of bisexual flowers, either stamen or carpel, it is suppressed completely and any one will be sterile. That one will be any stamen or carpel, either one will be sterile or it is suppressed completely. Okay. Or the flower may not be producing stamen, either stamen or carpel. That flower may not be producing either stamen or carpel. So cross pollination is must in that case because there is no pollen. See, in one flower which is having only stamen, there is no carpel part or it is suppressed, their cross pollination is must. There is no other option for that plant. So there is that should be a cross pollination. There is no other option for that. So suppression of one sex, that is the seventh method. In that bisexual flowers, either stamen or carpel, that is completely suppressed and any one will be sterile. Or either the flower may not be producing the stamen, either it may not be producing the stamen or the carpel. That also may happen. So there is no other option than cross pollination in such kind of flower. So suppression of one sex means either they won't be producing any one sex or one sex is completely suppressed in such kind of plants. Next, eighth method is heterostyle. In these type of plants, they may have two or more types of flowers. Heterostyle means different flowers, different types of flowers will be produced in such kind of plants. Okay. So these flowers have different length of style and stamens. In the stamen and style, they have different length. Okay. Heterostyle. Hetero means again different types. There is a difference in the style and stamens, in the length of style and stamens. That is called as heterostyle. So in oxalis, example, note down for heterostyle example, oxalis, 3 types of flowers are produced. So it is called as heterostyle. Heterostyle in the case of in the length of style and stamen. Okay, the difference is in the length of style and stamen. So there are 3 types of flowers in oxalis. That is called as heterostyle. Next, ninth method, in some plants, there will be some kind of physical barriers that may be present between anther and stigma. When it is coming, the anther and stigma, when it is coming near, there is a chance of self-pollination, right? So that plant itself prevents autogamy. The example, calotropis. Physical barriers are present in between anther and stigma. Example is calotropis. In calotropis, the pollen grains are present inside a sac-like structure. It is called as pollinium. It is called as pollinium. So a sac-like structure that is present in calotropis. Inside this only the pollens are present. It is called as pollinium, and mass of pollen grains are produced from a single anther. In this, so many pollen grains, a mass of pollen grains are produced inside this sac-like structure from a single anther. So there is a barrier between the anther and stigma. So in calotropis, due to the physical barrier like a sac-like barrier is present in calotropis. So the pollination is favored only with the help of insects. Only insects are helping for pollination because there is a barrier, a sac-like barrier between the anther and the stigmatic surface, right? So self-pollination, the pollen grains won't reach on the stigmatic surface. So there should be some pollinators or pollinating agents. So in the case of calotropis, with the help of insects, they are getting pollinated. So we have discussed around 9 methods. Which are the 9 methods of outbreeding devices? In that, there is no synchrony between the stigma receptivity and release of anther. That is the first method. Then anther and stigma are placed at different positions. The second method. Then self-incompatibility. The cell pollen grain of that same flower that is not suitable for the stigmatic surface. And production of unisexual flowers. In some plants, they are producing unisexual flowers to prevent the self-pollination. Then male and female flowers are located on different plants. Some plants will be producing only male flowers and some plants will be producing only female flowers. In the case of papaya, I said example for this is papaya. Then prepotency. Prepotency means that pollen grows faster from the pollen from different flower, another flower that will grow faster on the stigmatic surface compared to the pollen grains from the same flower. That is called as prepotency. Then suppression of one sex. Any one sex is suppressed in such kind of plants. And heterostyly means different types of flowers will be produced, which are showing difference in the length of style and stamen. Okay. Then some flowers, physical barriers will be present. Example for such flowers are calotropis. In that pollinium, which is a bag-like structure, inside that you may get so many pollen grains, which is produced from a single anther. Okay. So in that case, with the help of insects, the pollination is taking place. Okay. Next, we are going to discuss about pollen and pistil interaction. The interaction between the pollen and the pistil. We are going to discuss about this now. The pollen pistil interaction. That means that is a very complex process, and that starts from the acceptance of a compatible pollen on a stigmatic surface. I said the pollen and the stigmatic surface that should be compatible or that should be matching. Okay, then only that stigma will accept that particular pollen grain. See, you can see in the picture here 1, 2, 3, 4, 5, 6. 6 pollen grains are there. Correct? 6 pollen grains are there on the stigmatic surface. In that, the first one is not germinating. That may not be germinating due to some problem, or that may not be compatible with this stigmatic surface. It may not be matching for this stigmatic surface. And this is growing only up to this much distance. The second one, a very little growth is there. That means there are also some problems are there. Only the suitable pollen grain that will grow and the pollen tube will reach near the ovary. So there are so many reasons for all these things. Just now you have to learn this much only. The pollen grain and the stigmatic surface both should be compatible or matching. Then only that will lead to a fertilization. Okay. So the pollen pistil interaction that starts from the acceptance of a compatible pollen. The pollen grain should be compatible, then only the stigmatic surface is going to accept that pollen grain. So the interaction starts when it is getting accepted. From that time onwards, the interaction is starting. So the compatible pollen on the stigmatic surface reaching means from that onwards that interaction is going to start. And the steps which leads to a successful fertilization. So what is the pollen pistil interaction? When the pollen grain is landing on the stigmatic surface, if it is a compatible pollen, the stigmatic surface will receive or accept that pollen, and that pollen will germinate and it will lead to a successful fertilization. So all these interactions, they are together called as pollen pistil interaction, and such successful fertilization need a very clear pollen and pistil interaction. Okay, so how a pistil accepts or rejects that pollen grain? Do you have any idea? The chemical components of pollen of that pollen grain. You can see here, when the pollen is landing on the stigmatic surface, the chemical components, the pollen grain is having so many chemical components, and on the stigmatic surface also there are some chemical components. Okay, so the chemical components of pollen that interact with the receptive stigmatic surface, and that checks whether the pollen grain is compatible or not. So these interaction between the pollen grain, the chemical components of the pollen grain and the chemical components of the stigmatic surface, these interaction is taking place. The chemical interaction is taking place, and finally it is deciding whether this is a compatible pollen for this particular stigmatic surface or not. If it is not matching, it is getting rejected, and based on that the acceptance or rejection is taking place. Got it? And then if the pollen is compatible, if it is a compatible or matching pollen grain, the stigma accepts, that stigmatic surface will accept that particular pollen grain, and the pollen grain will start germinating on the stigma. Okay, so here you can see here 4 pollen grains are germinating, but only one is reaching to this ovary. Okay, why? So there are also so many reasons are there for that. Now in this chapter you have to learn only this much. The compatible pollen, only that pollen, there is the interaction between the pollen and the stigmatic surface. So the compatible pollen or the matching pollen will germinate, and it will reach or it will lead to a successful fertilization. And we have learned there are germ pores on pollen egg sign, right? While learning about the pollen grain structure, we have learned that there are germ pores on egg sign. So the pollen tube, that pollen tube grows through one of these germ pores. Through one of these, here you can see the exine and intine. We have learned about the layers of pollen grain, right? So that exine is having germ pores. Through any one of that germ pore, the pollen tube will start growing. Okay. So through this pollen tube, the content of that pollen grain that will move to the pollen tube. When the pollen tube is starting its germination, the content of that pollen grain from inside the pollen grain, that content will move along with that. That content will move into the pollen tube. Okay, and this pollen tube, it will germinate and it will reach to the ovule. Through how it is reaching the ovule? Through the micropylar region. While studying about the structure of that embryo or the TS of that flower, I explained about that micropylar region. Micropylar region, that is the opening. That integument is not covering completely. Integument is not covering completely this part. That leaves an opening. Through that opening, the pollen tube is entering inside the ovary. So now the male gamete and female gamete both are near to the ovary, right? So the pollen pistil interaction means the complex and cooperative process between the pollen and the receptive pistil from the acceptance of the compatible pollen and the subsequent steps which is leading to a successful fertilization. Got it? So the interaction between the chemical components, the interaction is taking place between the chemical components of the pollen and the pistil. And that interaction, after that interaction, it will decide whether to accept or reject the pollen. Okay. And the compatible pollen, it is followed by pollen germination. That pollen is germinating on the stigmatic surface, and that pollen tube will grow through the germ pore. Through the germ pore, that pollen tube will grow, and the content of that pollen grain, the whole content of that pollen grain will move to the pollen tube, and that content will enter to the ovule. With the help of that pollen grain, that content also will, pollen tube. That pollen tube, that content will enter to the ovule. Through which region it will enter inside the ovule? Through the micropylar region that will reach the ovary. Clear? And next, while studying the structure of pollen grain, we learned about the 2-celled and 3-celled condition of pollen grain. Hope you remember. 2-celled and 3-celled stage of pollen grain. Right? In 2-cell condition, there will be a large vegetative cell and a small generative cell. Hope you remember the structure of the pollen grain. In that structure, it is having a generative cell and a vegetative cell. The vegetative cell will be the large one, and the small generative cell also will be present. That generative cell will be floating in the cytoplasm of vegetative cell. Right? So in that 2-cell condition of that pollen grain, the generative cell means that small cell that will divide and it will form 2 male gametes. Okay, the generative cell will divide and it will form 2 male gametes during that pollen tube growth, during the growth of that pollen grain on the stigma. See, when it is landing on the stigmatic surface, the pollen grain is landing on stigmatic surface, it will start its germination. So at that time, there will be, if it is in a 2-cell stage, that time the vegetative cell and generative cell will be present. And when it is starting the germination, that generative cell, that means the small cell, it will divide and it will form 2 male gametes during the germination of that pollen grain. Okay. And if it is in a 3-cell stage, that pollen grain, if the pollen grain is in 3-cell stage, it will be having 2 male gametes. Already that will be having 2 male gametes. If it is in a 2-cell stage, the generative cell will divide and it will form 2 male gametes. Okay. So in such cases, the 2-cell pollen grain, generative cell will divide and form 2 male gametes during the growth of pollen tube in the stigma. Okay. And in the case of plants which are having or which are shedding the pollen grains in the 3-cell stage, that pollen tube already it will be having 2 male gametes from the beginning only. From the beginning itself, that 3-cell, if it is in a 3-cell stage, it will be having 2 male gametes. If it is in 2-cell stage, the generative cell will divide and it will form 2 male gametes. So after reaching the ovary, you can see after reaching the ovary, in the first diagram here you can see, now this is the pollen tube. See, this pollen tube is reaching here. Okay, near to the ovary. So this part we are zooming. This picture is that. The first diagram is the enlarged diagram of this part, the down part. Here you can see the pollen tube with one vegetative nucleus. The pollen grain, it is releasing the content into the pollen tube. So with that content, the pollen tube is reaching in the micropylar region, right? So it will be having a vegetative cell, the content of that vegetative cell and generative cell. Generative cell will divide and it will form 2 male gametes. Just now I discussed, it will form 2 male gametes. So here you can see 2 male gametes, and the vegetative cell, that nucleus will be released. So the vegetative cell nucleus, here you can see the vegetative nucleus plus 2 male gametes in the first diagram. Okay. So after reaching the ovary, that pollen tube, what the pollen tube will do, that pollen tube enters to the ovule through micropylar region. Through micropylar region, that pollen tube is entering into the ovule. And after that, it enters one of the synergates. Here you can see the synergates. It is reaching one of the, see here in micropylar region, 2 synergates will be there and one egg cell also, right? So it will enter one of the synergates through what? Through the filiform apparatus. The filiform apparatus. While discussing about this structure, that time itself I explained you. It is helping to guide the filiform apparatus. The function of that is guiding the pollen tube to reach the egg cell. Got it? So we have learned that filiform apparatus are guiding the entry of pollen tube, or that is a function of filiform apparatus. Don't forget, this is an important MCQ. You may get this question for NEET exam. So pollen pistil interaction is the event which are coming after the pollen deposition on stigma, right? On stigma, the pollen grain is getting deposited. So after that, all these events are happening. So until the pollen reaches the pollen tube, until that pollen tube is reaching the ovule, up to that, so many events are happening. The pollen is landing on that stigmatic surface. It is germinating. While germinating, that pollen grain is releasing the contents. It is releasing one vegetative nucleus plus 2 male gametes. So by carrying this vegetative nucleus and male gamete, that pollen tube is reaching near the micropylar region, and through the filiform apparatus it is entering. Then all these events together, all these together called as pollen pistil interaction. Got it? Do you have any doubt? So after learning the mechanism of pollen pistil interaction, what we can do? Why we are learning about this pollen and pistil interaction? This idea, this will help a plant breeder to manipulate the pollen pistil interaction, right? We can make some changes in the pollen pistil interaction, even in incompatible pollen grains also, right? See, I said some pollen grains are not compatible with the stigmatic surface, so it may get rejected. So with the help of this, we can learn why it is incompatible and what we can do to make it compatible, so that we can get desired characters, or we can make desired hybrids also by studying the pollen pistil interaction. Okay. There are so many things or so many events in this pollen pistil interaction. And in lab also we are doing this pollen germination experiment, right? In 12th standard practical examination, you will get pollen germination, one experiment. How you will do that? During the pollen grains, you can collect the pollen grains of a common flower, any hibiscus or something. We used to take the hibiscus flower pollen grains. We'll take that on a glass slide, and that glass slide will be having 10% of sucrose solution. With the help of that sucrose solution, you are going to do the pollen germination experiment, right? Why you are using sucrose germination? You are providing some nutrient for the germination of the pollen grain. So sucrose is a nutrient medium which is helping for the growth of that pollen grain, for the germination of pollen grain. So you are providing 10% nutrient solution or sugar solution, and on that you are going to dust this pollen grain. You are dusting these pollen grains on the surface of that nutrient solution, and you need to keep it for 10 to 15 minutes. And after 10 to 15 minutes, you can observe the slides under microscope. What you can see in the low magnification itself, you can see, or you can observe the tube-like something is growing. Tube-like structures are growing out of that pollen grain, that are called as pollen tubes. Those are the pollen tubes. Okay. So that's how you're doing the pollen germination experiment. Okay. So next is artificial hybridization. What is artificial hybridization? What is hybridization actually? Hybridization means The process of crossbreeding between genetically dissimilar plants. Why we are doing this? To produce hybrids. Hybridization, why we are doing? For the production of hybrids only. So hybridization, in between what we are doing? Genetically dissimilar plants. In the case of plants, we are selecting genetically dissimilar plants, or plants are having different characters, which we want to, those characters which we want to bring together in the next generation, we are going for hybridization. Okay. The process of crossbreeding, that is the hybridization. So crossbreeding or crossing, that will be done between different species to combine desirable characters, right? So that may get a superior variety. After hybridization, you may get a superior variety which is having the commercially important or commercially demanding characters. Okay, that will be having high demand. Commercially, that will be having high demand. So artificial hybridization, that is a crop improvement program. Artificial hybridization is a crop improvement program. So in such cross experiments or crossing experiments or hybridization experiments, we need to make sure that which pollen grain, which character we want, only from that plant we are collecting some pollen grains, right? And we are depositing on the stigmatic surface of particular plant, or which we are selecting plant A and plant B. Both are having some special characters. We want to combine both these characters into a single plant. Okay. So we are making sure that only that pollen grain with the different special character, we are collecting only those pollen grains, and only those pollen grains are getting deposited on the stigmatic surface. Okay. So that only that desired plants with the desired characters are getting produced, or only those pollen grains are reaching on the stigmatic surface. We need to make it sure that only those pollen grains are reaching, or the pollen grain is safe, or the pollination is safe, and the stigma is not contaminated with any other pollen grains. That we need to make sure. Otherwise, if all the pollen grains from different flowers are coming and sticking on the stigmatic surface, we can't make sure that our desirable character is there or not, right? So we need to make sure that we need to make it contamination free. Okay. So this is done by two steps. Artificial hybridization is done by two steps. First step is emasculation, and the second one is bagging. So emasculation means we are going to remove the anthers from a flower bud. From the bud stage itself, we will open the bud with the help of forceps, and we'll remove the anthers before the dehiscence of anther. If it is dehiscing, it will get self-pollinated, right? So before the dehiscence, before the maturity of that anthers, with the help of some forceps, we are opening those buds with the help of one needle. Then with the help of forceps, we are removing the anthers. You can see in the first diagram, we are removing the anthers by opening the floral bud. Okay. Then that emasculated flowers or the anthers are removed completely from that flower, and we have to protect these flowers from the unwanted pollen grains, right? So for that, that emasculated flowers, we are going to cover with some kind of bags, which is made up of butter paper and all. That procedure is called as bagging. You can see here. After emasculation, we are covering that particular flower with some polythene bag or some butter paper covers and all. For small flowers, we are using butter paper covers. So that is called as bagging. Okay. Then we can make sure that only desired pollen grains we are going to dust it as the next procedure. Then what we will do after bagging, then the desired pollen grains, we will collect the desired pollen grains from the anthers of a particular male plant, and we will dust these pollen grains on the stigmatic surface. Then we will do rebagging. After dusting these pollen grains, when we need to do rebagging, because we need to protect that particular flower from the pollination or from other pollen grains. Otherwise, some insects from the next flower and all, it will come and sit on this. Again, it will get contaminated with different pollen grains. We need pollen grains from a particular plant. So we need to do rebagging to protect. So artificial hybridization means that is having two steps, emasculation and bagging. Emasculation means removal of the anther from the flower or the floral bud. We will open the floral bud with the help of a needle, and before the maturation of the anther, we are going to remove the anthers completely. So there won't be any self-pollination. So we are preventing the self-pollination by this step. Emasculation is done. Next, the emasculated flower we need to protect from foreign anthers or foreign pollen grains, or from other pollen grains which is not having the desired characters. So for that purpose, we are going to cover it with particular covers or butter paper covers in the case of small flowers, or if the flower is big, or in the case of corn and all, you are going to cover with big polythene covers and all. That's how with bagging we are protecting the contamination or unwanted pollen grains. And after that, next day or after collecting the desired pollen grains, you are going to dust the desired pollen grains on the stigmatic surface, and you are going to do rebagging. Otherwise, again, it will get contaminated. Okay. So these are the steps of artificial hybridization. Is it clear? Clear? Hope it is clear. Next, double fertilization and some other topics are there. Then seed formation, embryo formation. All these things we'll discuss in the next class. So hope up to this everything is clear.
Clear, ma'am.
Okay, just go through this and go through the questions also. Just practice those questions. We'll see in the next class. You can leave now. Thank you. Thank you, students.